It is worth drawing a sharp distinction between the two modes of signal propagation in neurons. In dendrites, signals spread passively. The electrical disturbance fades with increasing distance from its source, like ripples in water weakening as they spread outward. This works well enough over the short distances involved in dendritic signaling. However, for the length of an axon, which can be a full meter long, passive spread would mean the signal would disappear entirely before reaching its destination. This is why axons generate self-amplifying action potentials instead. The action potential does not weaken because it regenerates itself at each successive point along the membrane, using the sodium and potassium channel machinery described in the previous section. Sodium and potassium pumps and membrane ion exchangers work continuously to restore the ionic gradients after each action potential fires, ensuring the axon can keep firing reliably. Without this restoration, the driving forces for sodium and potassium movement would diminish with each successive action potential. This distinction, passive propagation in dendrites, active regeneration in axons, reflects a fundamental architectural principle of the neuron. Information collection is passive and graded, allowing for summation and integration, while information transmission is active and all or none, ensuring fidelity over distance. Passive propagation involves current flow along the axon or dendrite membrane that passively makes adjacent segments less negative, insufficient for long-distance transmission as signal decays with distance. Relies on voltage-gated sodium channels opening once threshold is reached, generating new action potentials at each segment, preventing decay and enabling fast, long-distance signaling, with saltatory conduction contributing to increased velocity in myelinated fibers. Triggered when membrane potential reaches threshold, causing sodium channels to open and sodium influx to drive rapid upstroke. Potassium channels subsequently open to repolarize the membrane. Myelinated axons conduct impulses via saltatory conduction, in which local circuit currents depolarize nodes of Ranvier rapidly and efficiently, leading to much greater conduction velocity than in unmyelinated fibers. However, impulses do not jump as discrete events from node to node. Instead, active depolarization occurs simultaneously along substantial lengths of large axons. After depolarization, local refractory periods due to inactivated sodium channels ensure that action potentials only propagate forward, not backward. Disease states affecting myelin or ion channels, example demyelinating neuropathies, channelopathies, can significantly impair these processes.